Hello, everybody. We are going to talk about the second part of Chapter 3, which is actually a really important part of organic chemistry. We're going to look at conformations of acyclic alkanes and also conformations of six-membered rings, cycloalkanes, cyclohexanes. So for this section, I strongly recommend that you have a modeling kit on hand because making the models and playing around with the models are going to actually be very helpful in understanding this material. So let's get started. All right, let me get this off. So let's look at conformations. Remember, sigma bonds are formed by those head-to-head overlaps of orbitals, and they have unrestricted rotation. So any two groups that are bonded by a single bond can undergo rotation about that bond with respect to each other. This gives rise to a couple of terms that we're going to need to know. First, we have conformations, which is a temporary molecular shape that results from a rotation about the single bond. I did not break a bond or do any chemistry. I'm just simply rotating sigma bonds. And I can cycle through a variety of conformations just by rotating bonds. There's also the term conformer, which is a possible structure in that huge array of conformational possibilities. So if I took a snapshot of a molecule sort of at any point as I'm rotating the bond, each one of those snapshots would represent a different conformer of the overall molecule. Now, I can actually do an analysis of all of those conformer structures, and I could look at energy changes that occur as a molecule undergoes rotations, and I can assess stability. Is this a high energy, unstable conformer? Is this a low energy, more stable conformer? Based on how all of the different bonds and different substituents and atoms interact with each other in a particular conformer. So we're going to use something called a Newman projection in order to do this. What we want to do is start off with sort of a wedge and dash structure. Remember, a conformation, right? The conformer is going to be something that I'm looking for in three-dimensional shape, right? So I want to look at where my hydrogens are relative to another. And when I sort of rotate this, so I'm looking from, like if I have ethane here, if I sort of think about it in three dimensions, I go to there. But if this is carbon number 1 and this is carbon number 2, if this is carbon number 1 and this is carbon number 2, if I make the model, what I see is something basically called a sawhorse projection. And then what I'm going to do is I'm going to sight along this bond, and I'm going to look down the bond so that my carbons are right in back of each other in order to get to this Newman projection. Now I'm going to do this by showing you ethane and hopefully using the model. So I'm going to stop the share. Now if you look at the model, this is ethane, and notice I can rotate any of these bonds. So here's sort of my ethane, and then here's where I'm gonna make this sawhorse. I'm just sort of tilting it a little bit so that I'm looking at it where you're looking sort of down this bond. To make the Newman projection, what I'm gonna do is I'm gonna look straight from the carbon 1 to the carbon 2. And then I usually sort of put it where my front is staggered like this. And then notice this back bond. I'm gonna put this so you're looking at the front bond right here. And then notice the back bond can just rotate. And each one of these, if I stop here, 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 each one of those individual three-dimensional structures is actually a conformer. They're all ethane, only rotating around a single bond. Okay. Alrighty, so when I think about that, this is my Newman projection right here, and we're gonna be looking at these types of things. So what I'm gonna do, again, is I'm gonna pick 2 carbons in a structure. If I have 3, 4, 5 different carbons, I only look down one bond. So in this particular case, I'm looking down this bond. I'm sighting down, straight down that particular bond. My front carbon is going to be my perspective right here. And then I have this circle, and then directly behind my front carbon, I have my back carbon. And then this blue hydrogen are going to be the hydrogens on the back carbon, and the red hydrogens are the hydrogens on the front carbon. This is really hard to do, not in person. So the best way you can look at this is actually to look at building a model. So for example, I have this. Remember, this guy really looks like this, right? If I draw it out, if I cite, and this is carbon number 1, and this is carbon new, on carbon 1, I have 2 hydrogens and 1 methyl. On carbon 2, remember, I also have a hydrogen there. So I have 1 hydrogen, 1 methyl, and 1 chlorine. So I each have 3 groups, and this is going to be the second carbon here. So this is carbon 1. I have 2 hydrogens and a methyl. This is, behind that is carbon 2. I have a chlorine, a hydrogen, and a methyl. So that would be this guy. And I put them in this particular conformation where everything is sort of staggered from one another. I could draw it any way I see it. If I'm making a model, I'm just looking down the bond. And again, if I look at the conformations, I'm just going to keep the front carbon centered or not moving. And I can just rotate that bond and look at where the chlorine, the methyl, and the hydrogen of that back carbon goes around each one as I rotate that bond. So here's a question. Which of the following represents the Newman projection of the following structure that is drawn by sighting down the bond connecting carbons 3 and 4 and showing carbon 3? So again, this is a 1, 2, 3, 4, 5, 6 carbon hexane, but I'm only interested in how it looks from carbon 3 to 4. So if I look at carbon 3, I've got a hydrogen going down. I have this bromine coming at me. And then I sort of have that ethyl group there. Then in my sawhorse, I have that ethyl group coming down. I have that phenyl coming out towards me. And then I have the CH3 going backwards. So now here is where I'm looking. And this would be my 1, 2, 3, 4, 5, 6, that hexane carbon chain. So what I notice is that when I draw this on my front carbon, oops, I'm going to have the ethyl going on the top, the CH3 to the left, and the Br to the right. And then on my back carbon, I'm going to have the phenyl to the right, the CH3 to the left, and then the ethyl on the bottom. So this is what this Newman is going to look like. And basically what you see is I am going to have something that's going to look like this. Now remember, I can rotate it. This would be right, except these two are on the other side. I want the, this is the right one, right here. So sometimes you have to build it, and then you have to look at it, and you have to figure out what's happening. Actually, that's not right. This one is. I got myself around. If I just look at it, that's what I'm doing wrong. I'm looking at, this is actually a hydrogen off of here. So then this guy, I was trying to do too many things in the same point. So if I'm looking at this one, notice I have this hydrogen coming off, and then I have the bromine here. And I'm leaving this in the video because it's sort of, when I make a mistake, it's sort of, and then I talk through it, it sort of looks at how you should approach things when you're looking at it, right? You want to make sure you look at the carbon-carbon connectivity. You want to make sure that you get all of the groups that are attached to the carbon bonds that you're looking at. And what I had done was I made a mistake, which made it very difficult for me to look because I wasn't looking for the right structure. But this guy is what I drew based on the sawhorse, and that is actually the right answer. So let's talk about angles. As I rotate the bond, what I can look at is this angle here, the angle between the hydrogens, right? This theta, which is called the dihedral angle or the torsional angle. And that is showing me how far apart those two hydrogens are. Now, one of them is on the back carbon and one of them is on the front carbon, but they do form this dihedral angle, which is about 60 degrees in this particular conformer. This is called a staggered conformation because I have all of these dihedral angles here, where these hydrogens are pretty much as far apart from each other as they can be. And this is called staggered because these hydrogens are far apart from each other. They are considered to be more stable or lower in energy. This is eclipsed, and this eclipsed is where I have my hydrogens kind of stacked on top of each other. They're eclipsing each other, and that's going to be a higher energy. You can actually measure the difference in this energy, And the difference is due to torsional strain. I have a lot more interactions between these hydrogens than I have in this case, so that is not as stable. Remember, areas of electron density want to be as far away from each other as possible. And I can actually look at the difference in energy, and I can calculate that, and I can measure that. And between this staggered, if I was going to rotate and go to that eclipsed, there is a 12 kilojoule per mole energy difference between those two conformations. So I can actually look at this, and I can look at this rotation. So let me try to do this. And I'm going to try and put my screen up. So here I have ethane. I'm going to mimic so you see the bottom structure. So right now, I have got this structure right here. Front carbon, hydrogens pointing up, 2 hydrogens side. Back carbon, the 2 hydrogens are staggered, right, from this guy. And then I have back hydrogen on the bottom. So what I drew, what this conformation is actually indicating is right here on my screen. So I'm going to keep the front, and I'm going to rotate the back. And look what happens. Now I have eclipsed, right? These two are eclipsing each other. These two are eclipsing, they're close to each other. And what I did by doing that rotation is now I went to that eclipsed. Well then, and I went up in energy. Well, if I do another rotation, now I'm back down, oopsies. I have to get my pen. I'm back down here, right? Where now I have another staggered, and then I rotate it, and I get eclipsed, and then I rotate it, and I get staggered, and then I rotate it, and I get eclipsed, and then I rotate it, and I get staggered. Because each one of these is a hydrogen on all carbons, the eclipsed conformation and the staggered conformation are going to be the same when I look at rotating from this one to this one to this one. What I'm trying to say is that these 3 conformations, even though they're technically different from one another, are going to be equal energy. And then these guys are also equal energy. And the difference in energy is going to be that 12 kilojoules per mole. So this eclipsed is going to be 12 kilojoule per mole higher in energy than any one of the staggers. But all of these eclipsed are identical in energy because I have hydrogen-hydrogen interaction, hydrogen-hydrogen interaction, hydrogen-hydrogen interaction. So it doesn't matter which one of these conformations I use, they all have hydrogen-hydrogen interactions. Now, I can measure this, and what I know that the difference between the staggered and the eclipse is 12 kilojoules per mole. And I know that each interaction is between a hydrogen-hydrogen. So each one of these eclipsed interactions contributes 4 kilojoules per mole, right? The eclipse conformation is going to be less stable because of the electron pair repulsion between those eclipsing bonds. And that each one of those hydrogen-hydrogen eclipsing interactions is going to be worth 4 kilojoules per mole. There's 3 of them, in this ethane molecule, 3 times 4 is equal to 12. That's how I get the 12. With a difference like that, at room temperature, 99% of all ethane molecules are going to be in the staggered conformation. This is actually a pretty significant difference. Now let's look at propane. Propane has another carbon. All right, so I'm going to build propane, and I'm going to replace the carbon, or the hydrogen, with a carbon. So now I have 3 carbons, right? I have got my 3 carbon propane. I'm going to set it up so it looks like this guy right down here. So from your perspective, as I'm looking at this, I have the carbon number 2, right? So this is carbon number 1, 2, and then the 3rd carbon is in the back. Carbon number 1, I have the methyl group pointing up. I have hydrogen and hydrogen to the right and to the left. And then the back carbon, I have the 2 hydrogens back here, and they are staggered with the methyl. So this methyl is pointing up, then I have the bond, this hydrogen is pointing down. That's where I get where my dihedral angle between the methyl and the hydrogen is 180 degrees. Now, I'm going to keep the front constant. Actually, I'm going to keep the back constant, and I'm rotating the front so I can correspond to the diagram. I rotate. Well, now my methyl, right? This methyl group and the hydrogen are eclipsed. See, now I have this methyl and the hydrogen that are eclipsed. Then I rotate and I'm staggered. I rotate and I'm eclipsed. I have 2 hydrogen-hydrogen interactions and a methyl-hydrogen interaction. And then I rotate and I'm staggered. And then I rotate and I'm eclipsed. And I rotate and I'm staggered. What you're going to notice is in these ones, all 3 of these high energy conformations are also energetically equivalent. I always have 2 hydrogen-hydrogen interactions and 1 hydrogen-methyl interaction. And they're all equivalent because in every one of these 3 higher energy conformations, the interactions are identical. It's always 2 hydrogen-hydrogens and then 1 hydrogen-methyl. And again, here, these are all identical because these eclipsing or these staggareds are always moving the methyl group around. So, but now, because this interaction, this hydrogen-methyl, is going to be higher energy. And it's higher energy because the methyl is larger and my methyl takes up more space. It's going to be higher energy because it's going to run into those hydrogens more than if I had just the hydrogen-hydrogen interaction, right? Here's hydrogen-hydrogen, 2 small atoms. Here's methyl hydrogen. Well, now I've got this whole group interacting with my hydrogen. So this hydrogen-methyl interaction is much larger in terms of energy. So the barrier to rotation, to go from the staggered to the eclipsed, that's the barrier to rotation, is 14 kilojoules per mole. That's 2 kilojoules per mole than what we saw before, but you can explain it because of this. I have hydrogen and methyl group, which actually is 6 kilojoules per mole. So when I look at it, I have 4 plus 4 is 8 plus 6 is 14. Because the methyl group is larger, the hydrogen methyl eclipsing interaction is worse than, and raises the energy of the overall conformation. Now let's look at butane. where I start getting a little bit different of a situation. So now I'm going to take a methyl group and I'm going to add this, and now I have butane, right? Butane is going to be the model that I'm going to be looking at. I'm going to look down carbon 3 to 4. So now, if I look at carbon 3, so I have 1, 2, 3, 4. I'm looking down carbon 2 to 3. So on that carbon, let me draw this out. I have CH3, CH, H, CH, CH3. So I'm looking down this bond, which is carbon 2 to carbon 3. When I do that, I'm making the staggered, so I'm looking at this guy here, right? And what I'm going to do is I have a methyl group pointing up, and then the 2 hydrogens to the side, and then I have a methyl group pointing down, and then 2 hydrogens to the side. So again, I'm looking at this staggered. All right, I'm going to rotate the front by 60 degrees. Now I have the eclipse. Methyl hydrogen, methyl hydrogen, hydrogen, hydrogen. Okay, so I have the methyl hydrogen, and hydrogen, hydrogen. I have 2 of the larger interactions, and then I have 1 of the hydrogen, hydrogen, the smaller interaction. Now, let's look at this eclipsed. Uh-oh, I've got an eclipsing. Oh, before I do that, I want to look at this staggered. I want to look at this staggered. And notice I have got some interaction here because this methyl group and this bottom methyl group are really close to each other. Instead of being far apart, they're now closer. That's not as stable. So notice this, that staggered is higher in energy because I have a methyl-methyl gauche interaction. They're not eclipsing, but they're closer apart than when they were staggered. They're closer, they're closer together here. Now, let's go to the highest energy. Notice this eclipsing is higher energy than the other 2 eclipsed, and that's because that one has a methyl-methyl. This methyl-methyl, they are right on top of each other. So I have a hydrogen, hydrogen, a hydrogen, hydrogen, and then this methyl-methyl. This methyl doesn't want to be in the same space. And then I'm going to rotate. I have the eclipsing gauche on the other side. Then I rotate. I have the hydrogen methyl, the hydrogen methyl, and the hydrogen hydrogen. And then this is where I started from. So you need to look at the fact that as I rotate the bond, what I'm getting is differences in energies based on which groups are closer and farther away from each other. If bigger groups are closer together, that is a higher energy interaction, and it raises the energy of that overall conformer. So here, I have 2 different low energy conformations. I have the staggered anti, where my methyls are 180 degrees away from each other. And then at this level, I have the staggered gauche, where I have an interaction that's 60 degrees away from each other, but it's still staggered. Then I have two high energy eclipsing, but one extra high energy eclipsing where my two methyl groups are totally eclipsed over one another. So you need to be able to look at the model, look at the conformation, draw the conformations, relate the energy of the conformation to a situation like this where you have to do an analysis of what groups are closer together and what groups are farther apart in order to figure out which ones would be higher or lower in energy on a diagram such as this. You really want to use your models because it's very, very hard to visualize this stuff unless you actually can see it in 3 dimensions using your molecular models. So this is sort of a diagram of the Newmans. This is still butane, and this corresponds to the energy diagram we just saw before. Here I have the anti-conformer where my methyls are 180 degrees, and that's going to be overall my lowest energy. Here I have eclipsed, but I have a hydrogen methyl, and a hydrogen. Then I go to anti, but here I have this gauche interaction. So this is going to be higher in energy than 1, but lower in energy than 2, because this is still staggered. Then when I rotate, I get this very high methyl-methyl eclipsing. That's going to be the highest energy in terms of the higher energy eclipsed. Remember, staggered is higher energy than, or staggered is lower energy than eclipsed. But within the eclipsing conformations, as I rotate bonds, I could have an eclipsing conformation that's higher or lower in energy from one another. The same thing with the staggered. Here I have the absolutely lowest energy staggered where those methyl groups are far away from each other. And then I have these gauche staggered where, look at here, I have this other gauche interaction where this isn't as favorable as having them all the way 180 degrees from each other. And then again, when I rotate again, I'm looking at hydrogen, methyl, hydrogen, methyl, hydrogen, hydrogen. It's not as bad as that one. That one is the worst because I have those two eclipsing methyls. That's why that's the highest energy conformation there is. Now the stability of those different staggered conformations is about 3.8 kilojoules per mole. When those methyl groups are gauche, there is steric strain. Those, they're running into each other. Steric strain is like a spatial strain, right? They're in the same space. They're clicking into each other. That would be called steric strain. I have a higher steric strain, and that's going to be a higher energy conformation. When those methyl groups are anti to each other, when they're as far away from each other as possible, that has a lower steric strain, a lower steric interaction, and that is what I want to see. So I have these two conformations that are staggered that have equivalent energy, and this would be the lowest energy because I have this anti-positioning between those methyl groups. Again, if I look at the absolute highest energy conformation for the eclipsed conformations, it happens when these methyl-methyl is actually eclipsing each other, and that interaction results in 11 kilojoules per mole. So every methyl-methyl eclipsing interaction raises the energy by 11 kilojoules per mole. So this is the highest energy conformation for butane. All right, the next topic is going to be cycloalkanes, but I want you guys to think about these these conformational analysis with the cyclic alkanes, so we're going to stop here. And I'll be back. See you in a bit.